Session. This will be the first half of the Bushveld Complex session. And our first speaker for today would be Malte Junge. And over to you.
Yeah, thank you very much for the introduction. Well pronounced my name. And thanks for Steve and Freddie also for organizing this session and inviting me here to give the opportunity to give this very first talk of this Bushveld session. So back again to Bushveld and layered intrusions. During my talk, I will mainly focus on the reefs which are associated with particularly with the critical zone. So you all know this Bushveld Complex. Those of you who don't know, you have these stratigraphy, but in the critical zone, that's where we have all these chromatite seams. So I will talk about these and a bit about the UG2 chromatite and the Merensky reef, which occurs in the upper part of the critical zone. I will present a bit some work which was done in a bigger project where several PhD students were involved. The called Rammlmair project was coordinated by Bob Trumbull. And I will show some results of these projects as well. And they were dealing also the idea about whether it's possible to recover PGE as byproducts on these chromatites. So some slides from the chromite workshop. So some of you have seen this, but I just would like to still emphasize that these platinum group minerals, they still play a very important role when you talk about these reefs. Particularly when we have a look at the Merensky reef, there will be a lot of talks coming up about the Platte reef, also about this new or relatively new discovery of the flat reef, which I'm very looking forward for as well. But when you do reflected light microscopy images, you just see that they're always associated with these sulfides, which is most of the time the cases. You know this from different intrusions as well. But you also find them in rare cases associated with these chromite grains. And this is a particular case for laurite. So it's a ruthenium sulfide, which is mainly associated in the chromite. So this can be found in basically all these chromatite seams, including the chromatite stringers we have in the Merensky reef. What is interesting when you do automated mineralogy, so you have the opportunity to get really a lot of statistical data about the PGM distribution, way more than you can do with simple reflected light microscopy and looking through the samples, very tiny minerals, micrometer scale. We see that, and this is observed also for other studies on the Merensky reef and the Platte reef, that these are typically PGE bismutotellurides. And particularly when you look at the area percentage, which always good to do when you do automated mineralogy. So it's not simple frequency data, but it would just give you a more clear understanding about the entire PGE budget or in your ore. So you see that in the sulfidic ore, Merensky reef, Platte reef, these bismutotellurides are most abundant. There are other ones. Anna has shown us a few of these as well in her talk. But when you look at the chromatites, and this is one example from the Mg2 chromatite, you can see that you hardly find any of these PGE bismutotellurides. So these are mainly dominated there in these chromatites by PGE sulfides, PGE arsenites, and so on. So it seems that there's also some differences in the PGE mineralogy between these different ore horizons. So PGMs play quite important role for the PGE budget. So particularly when you look at palladium, palladium is the typical PGM mineral. But what is also known now since decades, basically describing that PGEs occur in sulfides, but we do see that there's some differences, particularly palladium and rhodium incorporated in sulfides. And when I talk about sulfides, this is particularly the case for pentlandite. So we see some kind of bimodal distribution, having PGMs, having PGEs in sulfide. Just a few words about this, because there will be also a talk on Friday by Jose Maria about nanoparticles. So I wanted to add this here as well, because the question then is, if you do these measurements, electron microprobe, laser ICP-MS, what is actually the signal you get? Is it the result of solid solution? So we have PGE just within the lattice, or do we have some very tiny inclusions of platinum group minerals which just give us a signal or the results in our database? When you do TEM, you can actually look at the nanometer scale, so 150 nanometer thin foils. You can look through the sample and you can see that we do have nanometer sized inclusions, 50 nanometer scale. These are just sitting in the pentlandite. So one reason, for instance, why we get our data is because we ablate or we measure some of these nanometer sized inclusions. But what's also true for the story is that when you look at these white patches here, and these are simple solid solutions. So in these cases, we have substitution with the PGE, with rhodium and nickel, in the case for the pentlandite. So both play a role, both are the results for what we see and forming these bimodal distribution. And I will come to this at the end of the talk as well. Last slide about this nanometer stuff. I still like it very much because it's very interesting to see this. When you looked at pentlandites having very high concentrations of rhodium, you will see that you observe these nanometer thin lamellae. And the advantage of the TEM, you can produce these very nice electron diffraction patterns. And you would see here this white dots, these reflections, which is the result of the pentlandite. And when you look careful, you will see that next to this, on between, you would see an additional reflection. And this is simply due to the case that when we have very high concentration, ordering takes place, and these PGE will occupy a very specific lattice site in the pentlandite structure, which is giving us this additional symmetry element, which we can then observe from a crystallography point of view. So that's probably the starting point of having the own PGM then at some point. What I also liked when we observed this study with the TEM is that you can also study larger PGM grains, so larger from a TEM point of view. So it's even a few micrometers, but you can still look through the sample, through these PGM assemblage and get this electron diffraction pattern. And what you see when you look careful in these higher orders of this pattern, these actually are two reflections just very close to each other. And the idea is that this could be the result because we had some embryos, precursors, however you would call them, but very early PGMs, which grew to each other with a slight misorientation, growing further and producing the slight shifted pattern. So something what Marianne Trédoux and others proposed already some decades ago would be some idea that this is also a contribution actually for early formed platinum group minerals in an early stage. Now moving to these chromatite seams in the lower and middle group. What I mentioned at the beginning, it was a project funded by the German Federal Ministry of Education and Research. But like I said, Bob Trumbull was coordinating this project. And what we were looking at this project is, we had a look at the Taba mine. So this is here in the western limb of the Bushveld Complex, northwestern limb, where they were mining these chromites for chromium production, basically. And we wanted to look if it's possible to get some idea about the PGE distribution and whether it's possible to process this as a byproduct. So there was a nice story about it. It was also nice because several PhD projects were involved, looking with different purposes on this project, but on a very nice mine scale. So we had drill cores covering quite detailedly the chromatite seams. Plus, we had the opportunity to sample within the pits, but also within trenches, which were just on the mine site, in order to also see what is the effect on near surface processes on these ores. Because of course, this is all mined open pit. So this might have an effect if we want to process PGEs from these ores, if we could also get good recoveries. One study was looking from Kai Bachmann, and he had a very nice data set because he was looking at all these primary drill cores covering LG-NG chromatites, and he was looking at the PGE mineralogy. So it was automated mineralogy, producing a large data set of platinum group minerals, and he could observe that there's a certain assemblage which is fitting these observation I showed you at the beginning. So we have typically PGE sulfides, which are associated with these base metal sulfides. So we have this typical assemblage we know from our primary ultramafic rocks. And with the automated mineralogy, of course, we get some information to what kind of minerals these PGMs are intergrown with. So we see that there are sulfides, chromides, silicates, what we typically observe. But what he also has shown is that there are also certain other composition of PGM mineralogy. And these are more dominated by PGE sulf-arsenides and the bit strange composition with PGE alloys, so antimony and bismuth. And these PGM mineralogy was also a bit different. It looked having associated rather with alteration silicates. And this is important information on the one hand for the general processes affecting the mine site, but also for beneficiation, because this then has an influence on the flotation procedure. If you would recover these by flotation, having these large amount of alteration silicates. Also the same idea, a recent publication by Joe, and together also with Bob Trumbull and Ilya and Kai Bachmann, who was also involved in this. And they were looking at a drill core, which was intersecting the UG2 chromatine. And this one was associated with these IRAP. So we heard about these IRAP during this conference, iron-rich ultramafic pegmatite. So this is actually a problem for a lot of these mining operations. And the nice thing is that they had the opportunity to really look detailedly on the intersections which are affected and which are unaffected by this IROP. And those which are unaffected, they typically show this normal PGE mineralogy, what we see. So typically, again, our PGE sulfides, laurite is ruthenium sulfide, and so on. So something what we observe from different studies all over the chromatite seams in the Bushveld complex. This does look a bit different when you look at these IRAP intrusions, because there you see that the mineralogy also changes. So you see a lot of these orange PGMs. So this is a PGE alloys, iron and tin. So the idea was also because it fits that also the mineralogy changes, the sulfide mineralogy, the millerite occurrence, and also now again a large abundance together with these iteration silicates. So also this has then effects on processing on the one hand, and it can also show us that these IRAPs, they do have quite some important implication also on the PGE mineralogy. So I mentioned there were various projects involved, and that's what I really liked about this project, to get to talk to the other people and focusing on these different observations. And we have seen some of these slides already in an earlier talk. In the last days, Felix Kaufmann, he was looking at the different chromatites and from a mineral textural point of view. And he has seen that these are these elongated oikocrysts, which are parallel to the layering. And so this work done together with Zoya and Marianne. And you could see that they have the same crystallographic orientation, but this in combination, looking at these kind of pyroxenes, they observed some differences in the mineral composition of the autopyroxene. So you've seen that some had relatively high enrichment of zirconium, rare earth elements, which are the core zones. And that's why the idea was that this is actually the result, which was formed by a peritectic replacement of the olivine by basically percolating melt, which was enriched in these incompatible elements and giving us this different mineralogical composition. we're also looking at the near surface stuff, as I mentioned at the beginning, because this might have some influence, because we observed this in previous work on the Platte Reef, on the PGE distribution. So one thing we did, what we can see here in red, this is a typical pattern you would see for this chromatite, typical shape, with the IPGEs and PPGs. And what is shown in the other colors is that these are samples taken either in the pit or in the trenches, or from near surface drill cores. And there you would see that actually the IPGE, they kind of overlap with the same pattern, but the rhodium, platinum, palladium, they are slightly decreased. So the idea is that because, as I showed you before, we have these laurite inclusions in the chromites, this is just protected by the weathering. So these won't be lost, similar like other works like the Chicoutimi group did, or Jung Park, about these IPGEs and chromites. So we also know that there is some contributions of IPG in the chromites themselves. So this is not mobilized. But we know that most of these plateau group minerals, that they are associated interstitial to these chromite grains, and they can then be more easily affected by weathering. So, and what we also did, we sampled the hanging and food wall. What you can see here, these gray circles, you can actually see that there is some concentrations, particular palladium, found in these hanging and food wall. So it shows us that there's some mobilization going on a very local scale, bringing us and reprecipitating some of the palladium and platinum in the surrounding silicate rocks. So we lose a little bit of palladium. That is what you can see here. So this line here shows the general slope, what you would have for platinum palladium ratio from this primary chromatites. And you see that the slope from the composition of these near surface chromatites is a bit with lower palladium numbers. So palladium is more mobile during weathering. We lose it, and that's why I would explain also the slope. But you can also see that the mobilization might be not go so far. It can also be ending up just at the hanging and food wall. Certainly, some extent of the palladium is just basically washed out and diluted, precipitating somewhere. But yeah, experimental work also shows that it's just more mobile. We also extracted some of the organic matter in the soils and had a look at humic acid fractions. And then we could actually see that this is also contributing or mobilizing some of the palladium. So organic matter also plays a role in transporting palladium for more palladium than platinum, at least. And also here we did some laser ICP-MS maps, and we could see that there's some correlation also with IPGE, with actually, or with platinum group elements in general, with a large different with a large group of other elements. And similar work was what we did on the Merensky Reef, where we compared sulfides, so the PGEs and sulfide. We see the same pattern again, what is known. We have blood group elements in the sulfides. But as these sulfides are easily oxidized, so we don't find them from 30 to 50 meters onward anymore. We see that using electron microprobe, for instance, but also laser ICP-MS, the PGEs are then hosted, for instance, in iron hydroxide, different silicates. So simply, yeah, different secondary silicates, basically. And so this is one contribution for these PGEs in these near surface ores. We also do still find some platinum group minerals. So some PGMs survive these weathering processes, but not all do. So I mentioned at the beginning, we have a lot of these PGE bismutotellurides in this sulfidic Merensky Reef ore. But when we look at the near surface, we don't find these anymore. We do find some others, but in particular, these bismutotellurides seems to be very unstable and they are just lost. But so this is a micrometer scale image of a BSE image in the MG chromatite. And what you can actually observe is that mobilization is also taking place on a very local scale. So here are some platinum group minerals, which were dissolved and then reprecipitated again along this small crack. So this is where we start to have the association with PGEs with the secondary silicates. So it's dissolving, but reprecipitating on the one hand on a very local scale, but also along mobilization in the hanging and food wall. And this brings us to the problem that we now, compared to this bimodal distribution we have in this primary ore, sulfide and PGMs, we have now a lot of different hosts for these PGEs. And this is a challenge for processing. So they try to process these ores, they receive recovery rates of less than 30%, which is of course not economic. And what we tried is to use a combination of sequential leaching with a large combination with bioleaching, with microorganisms. And there we could show that with a certain temperature range of the experiments combined with a certain type of microorganism, we could actually get recovery rates of this more than 90%. So maybe this combination with sequential leaching and microorganism might be an idea for processing these type of rock. On one hand, on these sulfidic rocks, like in the Platte Reef, but also what we have shown in the Tava mine, the Western Link. Coming a bit to a slightly different topic, but we have seen these kind of maps or kind of diagrams now quite often. And if you read about layered intrusions, you of course come across these very frequently. So a study Chris Tigner did some time ago on the upper zone and simply showing the distribution of the anortite content vertically. Simultaneously, what is happening with the clinopyroxene and magnesium number. We did some work some time ago on this UG2 chromatite, where we have seen that the chromite composition shows these different intervals, so different magnesium number, different chromium number. So there's probably rather some sublayers. Well, at the end, what I just want to show you here is that all this work does not matter whether it's a centimeter scale, decimeter scale, is very time consuming. So you need to prepare thin sections and electron microprobe usually. And the advantage of this new technology is like also what Wilhelm Nikonow was presenting and some time ago about these magnetite layers. And we have seen also these maps with Steve Barnes' group on these tornado or micro EDXRF. And I would like to spend a few words talking about these LIPS system, laser-induced breakdown spectroscopy. Because the nice advantage is that you can really scan entire drill cores or hand specimen and get some information on the major, minor, and partly also trace element composition. And the BGR in Hanover, with you might have seen the poster from Dieter Rammlmair, but also Wilhelm and Jeannet Meima, who have now like a very lot of experience with these methods and have a very good database about the mineral distribution. And so they are also good in evaluating this data, producing this raw material as this raw data. And so this is some work we did together looking at the distribution on the chromatites, where we could see where we tried to compare the electron microprobe work with the LIPS system. And what is actually shown here that it works quite nicely. So there's a relatively good correlation between the data we get from the chromite chemistry from the microprobe with those with the LIPS system. So this knowing, we had a look at the Marinsky Reef and had this almost 6-meter core where you could nicely see that you can produce this mineral distribution maps. So you can see here in blue, this is pleasure clays. So you can see the anortite, and then you would see that pyroxene jumps in, and here in yellow, you have the chromite and so on. So you can get some vertical variation about the distribution of your mineralogy. So this is already quite interesting, can tell you some interesting information. But the nice thing is that you get not only these informations, but also all the other informations combined with this. So whole rock analysis, some rough idea about the distribution of copper, nickel, potassium, and then you can use all this to correlate this with your mineralogy, because of course there's a link. But you can see here in brown, this is the phlogopite content. Here again, we have the chromatite. And the very nice thing, in my opinion, is that you can also get the information about mineral chemistry, because you can identify the different minerals and you get some compositional information, like was shown with the chromides. And for instance, here you can see there's a anotype variation, and you get it simultaneously to the variation, for instance, with the magnesium number and also pyroxene. So we get more or less a bigger picture, because all of these minerals, of course, compete about the composition and would help you to get the understanding of the formation. And for instance, here, Jeannet had this nice idea to also look, because you get all the statistical information, all this data, so you can also get an understanding what does the surrounding minerals do and what is their influence on the composition of these minerals. And this is shown here in this diagram, where you can see here phlogopides. This is all magnesium number and also pyroxene done by Lips. And you can see those which are colored in black are dominated, surrounded by phlogopite, those in blue by plagioclase, and those here magenta and pink by also pyroxene. And you can immediately see that, for instance, those where you have phlogopite dominant mineralogy, we also see a difference in the magnesium number and also pyroxene. Same here, just some also pyroxene variation. This is actually not too much influence in the magnesium number, but with the plagioclase, but there's still some differences. And you can nicely visualize this and see this and compare this to your whole rock data, to the mineralogy distribution, to get some understanding also about where we have the PGE enrichment. How do we get this PGE, for instance, in the system? What is the association also regarding the phlogopite and so on? Also here, you can play around with different ratios if you like. This is, for instance, shown the ratio of also pyroxene to kleiner pyroxene. And all this in combination would then help to get a better picture on the formation. So here, just some other examples, plagioclase, CPX ratio, and so on. So I think this is just a very powerful tool, particularly when having this big database, what they collected now, scanning these cores and getting more detailed information on the micrometer scale, and combining this with these observations we have. On this last slide, I would just like to talk about a very specific occurrence in the Eastern Bushveld, where we have these dunitic pipes. There's, for instance, this one here in the Onverwacht, so very interesting shape, and also very interesting PGE mineralogy. So these are typically very platinum dominated. So most of these PGMs are very rich in platinum, which you can see also here on these chondrite normalized pattern. And these platinum minerals, this mainly platinum iron alloys, you can see that they are actually quite big and associated with these chromite grains. Also, these are different PGMs, so all these different colors represent the different platinum group minerals. One idea was that this is just a result of upward penetration of magma replacing the cumulates. So these would produce a very iron-rich melt, because all these dunites are very iron-rich, autonolytic, so having very special composition. And this brings me to my final slide, and would just like to emphasize again what we also have seen in various other talks regarding these core scanning facilities. And I think it's a very nice method in combination with field observations, when we have seen these nice pictures always from Rais and others. But combining this with a more detailed work on the micrometer scale on the different drill cores, but rather than selecting with electron microprobe, time-consuming work, and combining this with more statistically work and forcing ourselves to not look at the specific stuff what the eyes wants to look at, but rather letting the machine as well finding these observations or the similarities. So this is, I think this LIP system is very interesting also for exploration and or mining and processing. And the last statement is just regarding these near surface ores. So mineralogy is really the key. So important to look at the mineralogy to see what is hosting these PGEs in order to come up then with a method, a processing technique, how we could make these rocks to an ore and make it economically available. And yeah, with this, I would like to end, and happy if there are any questions.
Just a very small question. Thank you for your talk. Which process can, from your opinion, fractionate platinum from palladium? Maybe I missed it. Like, what is, in your opinion, responsible for this spike of platinum?
For the platinum in these pipes. That's a good question. There are different studies about this. It's also something, there are no sulfides involved, so there are also some processes with desulfurizes, and it's certainly something what is different to what we have for the normal formation of PGEs from sulfide. So yeah, rather some more unique processes. But I think there's no, I don't have a clear answer to this.
Thank you.
Great.
Thank you. Very nice and interesting observations about that you told secondary platinum mineralization in chloride, for example, on this slide. But the question is, we often have such mineralization in secondary silicates when these silicates just replace sulfides in the primary assemblage. So how do you think how we can distinguish the truly reprecipitated platinum minerals from those who just reside while sulfide is replaced?
So I do think, so one opportunity, of course, is to look at the elements surrounding this. So some of these are still quite having some nickel concentrations, would be then showing that it's like former pentlandite and so on. But I think it's quite tricky because on the one hand, the PGEs and sulfides on the one hand are very small, and these platinum group minerals are still associated with these sulfides.
Yes, that's a problem. We discussed it for the Norals type and